Right, so I'm a clinician, so I'm going to speak very plainly, and we're going to zoom out a little bit from all that detailed T-cell work into something a little bit more clinical. Right, so I'm going to talk about the host determinants, actually going one step back from the adaptive immune responses of vaccine responses and focusing on T-cells and using a pediatric cohort. Okay, so I introduce you to this cohort, markers of vaccine efficacy and longevity in SARS-CoV-2. This is a cohort of healthy 5 to 12-year-olds getting primary vaccination during the pandemic. And just to note, because this was done in Singapore, really a lot of, most of the children were SARS-CoV-2 infection naive at that point, and we excluded any children who had been pre-infected. So just to lay the ground for the work that I'm going to present today, I will show you our previous published data, which really was to discover the correlates of protection against symptomatic SARS-CoV-2 in this cohort. And using a longitudinal cohort study design, as well as serial measurements of immunological parameters, so antibodies, T-cell responses, as well as memory B-cell responses along the way, we were able to, using this design, discover that, that the correlates of protection against symptomatic SARS-CoV-2 infection included binding antibodies, so anti-spike IgG levels, not neutralizing antibodies, whether it's against Wuhan Hu1 or BA2, spike-specific memory B cells, as well as T-cell responses. And this was measured, the T-cell responses were measured with a cytokine release assay on stimulation of whole blood with overlapping peptides against spike. And we were able to develop a model, a new paradigm of thinking about correlates of protection from our work. We call it the onion model because we think it works in layers, where the outermost layer would be neutralizing antibodies, and failing then the core protection was provided by T-cell responses. Now, the strength of prediction of the T-cell responses was really very significant in our cohort. So we were able to show using a receiver operating curve that, in fact, the area under the curve was 0.75, which is as good as it gets for a clinical biomarker to predict a clinical outcome. And so T-cell responses were really the most important predictor of risk for symptomatic infection with SARS-CoV-2 in vaccinated children. So now, if T-cell responses are very important, then we want to go one step back to really see what are determining these responses. So we know that the protection that is provided by vaccines is really mediated through adaptive immunity, which lasts months to hopefully years, which is provided both by antibodies and T-cells. But what then determines the quality and potentially longevity of the adaptive immunity is really that innate immune response, which occurs minutes to days after vaccination. And the innate immune response is important because it both provides the inflammatory milieu for adaptive immunity to take place, to be initiated, and of course presents antigen to the adaptive immune system. And so there's been a whole body of work using systems vaccinology methods that have discovered gene signatures and immune endotypes that predict antibody responses later on. But in the context of T-cell responses, which we now know are important for protection, at least in this cohort, is very much unknown. And so back to the Marvels cohort, we then went back to the cohort and selected the children that remained uninfected with SARS-CoV-2 from vaccination up to 3 months post-vaccination. And of course, again, just to emphasize, these children had no clinical history or serological or even T-cell evidence of natural infection, whether symptomatic or asymptomatic. So we used bulk RNA sequencing at baseline, as well as day 1 post-vaccination, where it's been shown that it's the peak of innate immune responses, to discover gene signatures that then predict spike-specific T-cell responses. So going back to the ROC curve, we selected the 80% sensitivity threshold as a cutoff for T-cell responses, which then helped us to segregate our children into the high responders and the low responders. And between the high and low responders, now the group of that were considered high responders had about a 7-fold increased T-cell responses at month 3 post-vaccination compared to the low responses. Now, comparing the fold difference of day 1 normalized on day 0 gene expression, we were then able to discover 86 genes which had higher magnitude of fold difference at day 1 in high responders, and about 180 genes that had higher magnitude of fold difference at day 1 in low responders. Throwing these 86 genes into an unsupervised clustering, we confirmed that the genes were actually very good at differentiating between low and high responders, misclassifying only one subject. And among these genes, we then discovered 3 specific biomarkers. So this is leptin, this is phospholipase C epsilon 1, this is phospholipid phosphatase. These were really strong predictors of poor T-cell responses, so downregulation of them predicted poor T-cell response. Again, using an ROC, you can see that the area under curve, when you combine the 3 biomarkers, was almost 0.9. In the lack of material that we could then drill down on the proteomics or the immunophenotype at that time point, what we did to then try and tease out what these genes were doing was to look for gene modules that were co-regulated with these 3 genes. So using that method, we were then able to discover lots of gene modules that were co-regulated with these 3 genes, but what was really striking was that what was common in terms of co-regulated gene modules with these 3 important biomarkers were all in the innate immune response, innate immune system. And drilling down there, you can see that the gene modules were in activation of dendritic cells, in viral sensing, and in interferon response. And taking a network view at these gene modules, you can see that a lot of the leading edge genes are important hubs in the innate immune response and all very interconnected. And specifically, master transcription factors such as STAT1, IRF7, and ATF3 were very important. Specifically looking at these 3 transcription factors, you can see that there was a much higher expression an upregulation of these genes at day 1 post-vaccination in the low responders for STAT1, IRF3, and ATF3. And so putting it all together, to summarize, in the low T-cell responders, you can see that there is a significant downregulation of these 3 genes, so the leptin, the PLCE1, the PLPP2. This was associated with what we now think is an excessive innate immune response mediated through ATF3, STAT1, IRF7, associated with a lower T-cell response at month 3 post-vaccination, which we now know is strongly associated with increased risk of infection. The high T-cell responders, on the other hand, there was no significant downregulation of these biomarkers associated with what we now think is an appropriate innate immune response and a stronger T-cell response at month 3 post-vaccination, which we now know is protective against symptomatic infection prospectively. Interestingly, that was, this finding is also associated with our other finding that when we compared the children to adults, significantly these children received a 1/3 reduced dose of vaccine, so that's 10 micrograms compared to adults who we all received 30 micrograms. This is the Pfizer BNT162b mRNA vaccine. And at 3 and 6 months time points and later time points as well, we saw that children consistently had stronger T-cell responses, and whether it could be explained by the excessive inflammation that is caused by the higher dose of mRNA vaccine, which we know now is hyper-inflammatory. We're not sure yet. And in our future work, so we've done this part, so we're going to go on to look at pre-vaccination immune endotypes, so including gene expression and immune endotypes as well, to see if that also explains some of the variants that we see in the all-important T-cell responses at month 3. And I'd like to acknowledge the team that made this happen, so specifically the co-investigators Elizabeth Tham and Lynette Steck, as well as the CRCs and research assistants who did all the work. My mentor, Eng Yong, who's I think here in the audience still. Yes. And Guan Rong, my collaborator, who did all this bioinformatics work. And of course, all the wonderful, marvelous children and families who were really the real heroes in the study and during the pandemic. Thank you.
So very lovely work. I was just wondering, had you looked at the ages of the children and seen if there was any correlation with the low responders being the younger of the children, and what was the age distribution?
Yeah, so this was actually quite a narrow age range, so 5 to 12-year-olds, so neither preschoolers nor adolescents in the primary school kind of age group. There was no significant correlation in terms of T-cell responses and age. Yeah, in fact, no demographic predictors that we could find of the T-cell responses.
Because we have some data we're just about to publish, but in a mouse model where we're looking at neonatal immunisation. And I think essentially we've found the same thing. We were immunising with a flu antigen with CpG, which is obviously an inducer of interferon responses. And we don't see any protection in the neonates, whereas it works in the adults. But when we actually gave the same vaccine to type 1 interferon knockout neonatal mouse, suddenly the vaccine was protective. So in other words, it's the interferon response that's only occurring in the neonates because they are hyper-responders with type 1 interferons, that appears to suppress the adaptive immune response. So it's just fascinating to see maybe the mouse and the humans correlate, which doesn't always happen.
Interesting. Thank you. Thanks for sharing that.
So Youjia, great talk here. Yeah. But similar question. But now you show that the innate response. It seems that almost all of them are type 1 interferon response.
Yes.
Then what do you think? The type 1 interferon directly affect CD8 T cell or the T cell, or indirectly through other cell?
Yeah, I think that's. I think we just don't have the ability to answer that question, and it's a really good question. So I think the first thing to say is that our assay doesn't differentiate between CD4 and CD8 T cells, and well, in our collaborative work, hopefully we can tease that out. Yeah, so I think first you need to probably see whether it's the CD8 or the CD4 T cell responses. What would you say to that question?
Oh, actually, so I think that my hypothesis is direct effect, because the type 1 interferon somehow the anti-proliferative effect, so maybe in T cell. So maybe that's the reason why. So, yeah.
So maybe CD8, you mean?
Oh, yeah, CD8 both. Both.
Both,
yeah,
okay.
Yeah.
Okay, maybe both.
Thank you.
Thanks.
Very nice talk. Just a very naive question. Do you see any long COVID symptoms in the pediatric cohort?
Not in this cohort, yeah, and it's kind of less common in children, yeah, and particularly in this age group. I think slightly more common in the adolescents, yeah.
Nice talk. So have you see the elderly people, if they have higher innate immunity, like people more more than 60 years old?
Yeah, that's an interesting question. So we haven't done those experiments, but what I would just hazard a guess at is that I think the immune senescence that actually is responsible for the blunted vaccine responses in the elderly is a little bit different, yeah, from the mechanisms that are there in healthy children. But it could be that somehow there's a dysregulated innate immune response that is part of that immune senescence, yeah, that leads to blunted vaccine responses.
Thank you.
lots of work to do.